Welcome back, class. Today's lecture is Lecture 7, Control of Microorganisms by Physical and Chemical Agents. So in this lecture we'll be focusing on the control of microbes, but keep in mind that there are lots of mechanisms used to control microbes that are not covered in this lecture. So for example, the manipulation of environmental conditions is used extensively in the food industry to preserve foods. Increased solutes such as sugar and salt are used to preserve meats and jams, for example. Microbial fermentations of milk and vegetables decrease the pH of these foods, and this creates new foods such as yogurt and cheese and so on, and all of these have a longer shelf life than the milk and vegetables from which they're made. Heat and the generation of anoxic conditions are also important in preserving canned foods, and ionising radiation is used to extend the shelf life of seafood, fruits and vegetables. Now we're going to take a look at the use of physical methods in microbial control. The four most frequently employed physical agents are heat, low temperatures, filtration and radiation. Heating is still one of the most popular ways to destroy microorganisms. Either moist or dry heat may be applied. Moist heat will readily kill viruses, bacteria and fungi. And moist heat is thought to kill by degrading nucleic acids, by denaturing enzymes and also denaturing other essential proteins. It may also disrupt cell membranes. Now, exposure to boiling water for 10 minutes is enough to destroy vegetative microbial cells and eukaryotic spores, but unfortunately the temperature of boiling water is not high enough to destroy bacterial endospores, which can actually survive hours of boiling. In order to destroy endospores, moist heat sterilisation has to be carried out at temperatures above 100 degrees Celsius, and this requires the use of saturated steam under pressure. Steam sterilisation is carried out using an autoclave, which is just a device much like a large pressure cooker. The development of the autoclave by Chamberlain in 1884 tremendously stimulated the growth of microbiology. Using an autoclave, water is boiled to produce steam. This is released through the jacket and into the autoclave's chamber. The air initially present in the chamber is forced out until the chamber is filled with saturated steam, and then the outlets are closed. Hot saturated steam continues to enter until the chamber reaches the desired temperature and pressure. This is usually 121 degrees Celsius and 15 pounds of pressure. At this temperature, saturated steam destroys all vegetative cells and endospores in a small volume of liquid within about 10 to 12 minutes. Treatment is continued for a further 15 minutes just to provide a margin of safety. This table summarises the approximate conditions for moist heat killing of different organisms. So we have listed here yeasts, molds, bacteria and viruses. But note also the difference in time and temperature required to kill the vegetative cells of each group of organisms in comparison to the spores produced by those organisms. Here we have an example of a modern automatically controlled autoclave or steam steriliser. To the right you can see a cross-section of a typical autoclave showing some of its working parts and also the pathway of steam. Many substances, including milk, are treated with controlled heating at temperatures well below boiling, and this process is known as pasteurisation in honour of its developer, Louis Pasteur. In the 1860s, the French wine industry was plagued by the problem of wine spoilage, which made wine storage and shipping very difficult. Louis Pasteur examined spoiled wine under the microscope, and he detected microorganisms that look like the bacteria responsible for lactic acid and acetic acid fermentations. He then worked out that a brief heating at 55 to 60 degrees Celsius would destroy these microorganisms and preserve wine for long periods. Then in 1886, the German chemists adapted the technique for preserving milk and reducing milk transmissible diseases. Milk pasteurisation was introduced into the US in 1889. Milk, beer and lots of other beverages are now pasteurised. So keep in mind that pasteurisation does not sterilise a beverage, but it does kill any pathogens that are present and drastically slows down spoilage by reducing the level of non-pathogenic spoilage microorganisms. Many objects are best sterilised in the absence of water by dry heat sterilisation. And this involves placing items in an oven at 160 to 170 or 180 degrees Celsius for 2 to 3 hours. Microbial death apparently results from the oxidation of cell constituents and denaturation of proteins. Keep in mind that dry heat is less effective than moist heat. For example, the endospores of Clostridium botulinum, the cause of botulism, are killed in 5 minutes at 121 degrees Celsius by moist heat, but only after 2 hours at 160 degrees Celsius with dry heat. However, dry heat has some definite advantages. It doesn't corrode glassware and metal instruments, as moist heat does, and it can be used to sterilise powders, oils, and so on. Most laboratories sterilise glassware in pipettes using dry heat. But despite these advantages, dry heat sterilisation is much slower than moist heat, and it's not suitable for heat-sensitive materials like many of the plastic and rubber items. Some items can be sterilised by dry heat incineration. For example, inoculating loops, which are used routinely in the laboratory, can be sterilised in a small benchtop incinerator, as pictured here. So we've seen that heat is very useful in controlling microorganisms. So it's essential to have a precise measure of heat killing efficiency. Initially, effectiveness was expressed in terms of the thermal death point, TDP, which is the lowest temperature at which a microbial suspension is killed in 10 minutes. But because this measure implies that a certain temperature is immediately lethal, despite the conditions, thermal death time, or TDT, is now more commonly used. This is simply the shortest time needed to kill all organisms in a microbial suspension at a specific temperature and under defined conditions. But remember that heat killing is logarithmic. So an even more precise figure, called the decimal reduction time, or D value, has gained wide acceptance. This measurement is the time required to kill 90% of the microorganisms or spores in a sample at a specified temperature. And we can see that in this logarithmic plot of the population remaining versus the time of heating. And we saw this on one of the previous slides. So the D value is the time required for the line to drop by 1 log cycle. And you can see that the D value written here is written with a subscript, which indicates the temperature for which it applies. And we can see D121 equals 1 minute. That is, 1 minute is required, the time required for the line to drop by 1 log cycle. D values are then used to estimate the relative resistance of a microorganism to different temperatures through calculation of the Z value. The Z value is the increase in temperature required to reduce the D value to 1/10 of its value, or to reduce it by 1 log cycle when log D is plotted against temperature. So in this example of a test microorganism, the Z value is shown here as 10.5 degrees Celsius. Let's look at how the food processing industry makes extensive use of both D and Z values. So after a food has been canned, it must be heated to eliminate the risk of the disease botulism, which arises from endospores of the bacterium Clostridium botulinum. Heat treatment is carried out long enough to reduce a population of 10 to the 12 Clostridium botulinum spores to 10 to the 0, that is 1 spore. Thus, there is a very small chance of any can having a viable spore. So let's look at the D value for these spores at 121 degrees, and you can see that the D value is 0.204 minutes at that temperature for Clostridium botulinum. Therefore, this tells us that it would take 12 times D, or 12 times 0.204, which equals 2.5 minutes, to reduce 10 to the 12 spores to 1 spore by heating at 121 degrees Celsius. The Z value for C. botulinum spores is 10 degrees Celsius, which you can see in the far right column. So this means that it takes a 10 degrees Celsius change in temperature to alter the D value tenfold. So if the cans were to be processed at 111 degrees Celsius rather than at 121 degrees Celsius, the D value would increase by tenfold to 2.04 minutes, and the 12D value to 24.5 minutes. Often the most convenient control technique is to inhibit the growth and reproduction of microbes by using either freezing or refrigeration. This approach is particularly important in food microbiology. Freezing items at -20 degrees Celsius or lower stops microbial growth because of the low temperature and the absence of liquid water. Some of the microorganisms will be killed by ice crystal disruption of cell membranes, but freezing doesn't destroy all contaminating microbes. In fact, freezing is a very good method for long-term storage of microorganisms, particularly when carried out properly. And many laboratories have a low temperature freezer for culture storage at -30 or -70 or -80 degrees Celsius. So refrigeration greatly slows microbial growth and reproduction, but it doesn't stop it completely. Fortunately, most pathogens are mesophilic and don't grow well at temperatures around 4 degrees Celsius. But refrigerated items may still be ruined by growth of psychrophilic and psychotrophic microorganisms, particularly if water is present. So refrigeration is a good technique only for short-term storage. Filtration is an excellent way to reduce the microbial population in solutions of heat-sensitive material, and sometimes it can be used to sterilise solutions. Rather than directly destroying the contaminating microorganisms, the filter simply removes them. There are two types of filters: depth filters, which consist of fibrous or granular materials that have been bonded into a thick layer, and it's filled with these tiny twisting channels of very small diameter. The solution containing the microorganisms is sucked through this layer under vacuum, and the microbial cells are removed by physical screening, entrapment, and also by adsorption to the surface of the filter material. These filters are made commonly from substances such as diatomaceous earth, unglazed porcelain, asbestos, or other similar materials. Membrane filters have largely replaced depth filters for many purposes. These filters are porous membranes, a little over 0.1 millimetres thick, and they can be made of cellulose acetate, cellulose nitrate, polycarbonate, or other synthetic materials. So although there's a wide variety of pore sizes available, those membranes with pores about 0.2 microns in diameter are commonly used to remove most vegetative cells, but not viruses. The membranes are held in special holders. The solution is pulled or forced through the filter with a vacuum or with pressure from a syringe, or a peristaltic pump, for example, and collected in sterilised containers. So membrane filters can remove microorganisms by screening them out, much like a sieve separates large sand particles from small ones. These filters are used to sterilise pharmaceuticals, culture media, oils, antibiotics, and any other heat-sensitive solutions. In A, you can see the bacterium Bacillus megaterium being caught on a nylon membrane. And this membrane has a bacteria removal rating of 0.2 microns. This image is at times 2000 magnification. The image to the right shows the bacterium Enterococcus faecalis resting on a polycarbonate membrane filter with a pore size of 0.4 microns. And this image was taken at times 5900 magnification. The photo shown in A shows a complete filtering setup. The non-sterile solution is in the Erlenmeyer flask, labelled 1. The peristaltic pump, labelled 2, forces the solution through the membrane filter unit, which is labelled with the number 3. In B, we can see a schematic representation of a membrane filtration setup that uses a vacuum pump to force liquid through the filter. The inset shows a cross-section of the filter and its pores, which are too small for microbes to pass through. And in C, we can see a cross-section of a membrane filtration unit. Several membranes are used together to increase its capacity. Air can also be sterilised by filtration. Two common examples are surgical masks and cotton plugs on culture vessels. These cotton plugs let air in but keep microorganisms out. Other important examples are laminar flow biological safety cabinets, which use high efficiency particulate air filters. In other words, HEPA filters. And these are a type of depth filter that are used to remove 99.97% of 0.3 micron particles. Laminar flow biological safety cabinets, otherwise known as hoods, force air through HEPA filters, then project a vertical curtain of sterile air across the cabinet opening. So this protects the worker from microorganisms being handled within the cabinet, but also prevents contamination of the room. So people use these cabinets when working with dangerous biological agents such as Mycobacterium tuberculosis and tumour viruses. They're also employed in research labs and industries when a sterile working surface is needed for conducting assays, preparing media, examining tissue cultures, and so on. Here we have a schematic of a laminar flow biological safety cabinet, and you can see the location of the HEPA filter and also the high velocity air barrier that's created. Microbiologists can also take advantage of the effects of ultraviolet and ionising radiation to sterilise or disinfect objects. Ultraviolet radiation, around 260 nanometres, is quite lethal to microorganisms, but it doesn't penetrate glass, dirt films, water and other substances very effectively. So because of this disadvantage, UV radiation is used as a sterilising agent only in a few specific situations. UV lamps are sometimes placed on the ceilings of rooms or in biological safety cabinets to sterilise the air and any of the exposed surfaces. But because UV radiation burns the skin and damages eyes, people working in these areas have to be certain that the UV lamps are off when the areas are in use. Commercial UV units are available for water treatment. Pathogens and other microorganisms are destroyed when a thin layer of water is passed under the lamps. Ionising radiation is an excellent sterilising agent and penetrates deep into objects. It will destroy bacterial endospores and vegetative cells. However, it's not always effective against viruses. Gamma radiation from a cobalt-60 source is used in the cold sterilisation of antibiotics, hormones, sutures, and plastic disposable supplies such as syringes. Gamma radiation has also been used to sterilise and pasteurise meat and other food products. This type of irradiation can eliminate the threat of pathogens such as E. coli, Staphylococcus aureus, and other pathogenic microbes. Based on several studies, both the Food and Drug Administration and the World Health Organization have approved food irradiation and declared it safe. Currently, irradiation is being used to treat poultry, beef, pork, veal, lamb, fruits, vegetables, and spices.